Acid-base equilibrium. Titration curves involving weak acids and weak bases. In this tutorial, we will be going over titrations involving a weak acid and a strong base, titrations involving a weak base and a strong acid, sketching a titration curve, weak base and a strong acid, sketching a titration curve, weak acid and a strong base, and finally, titration of a weak diprotic acid with a strong base. Titrations involving a weak acid and a strong base. When a weak acid is neutralized by a strong base, the graph varies in a few ways. First, the equivalence point does not equal a pH of 7. Instead, it's going to be greater than a pH of 7. So here in the graph, we can see that the equivalence point is at a pH of 9. A buffer region exists as the equivalence point is approached. The half equivalence point is when half of a weak acid is neutralized. So the equivalence point here occurs at 25 milliliters, where the concentration of the weak acid is completely neutralized. Half equivalence occurs at 12.5 milliliters, where half of the weak acid is neutralized. For weak acids, at the half equivalence point, the moles or molarity of the weak acid is equal to the moles or molarity of the conjugate base. Therefore, the pH at half volume of the equivalence point, otherwise known as half equivalence, is equal to the pKa for the acid. So in other words, if we look at the Henderson-Hasselbalch equation, this section where log is equal to the concentration of the weak base divided by the concentration of the weak acid, these two values are equal to each other, so they're going to cancel out. That is why pH is going to equal pKa. The area around the half equivalence point is called the buffer region, since the weak acid and the weak conjugate base are both present in solution. At volumes less than the half equivalence point, the non-ionized acid HA is the primary species. So we can see that on the graph right here, where the concentration of HA is larger than the concentration of the weak base. At volumes greater than half equivalence, the conjugate base, A-1, is the dominant species. So past half equivalence, the concentration of the weak base is going to be greater than the concentration of the weak acid. They both exist in this region, but there's more of the weak base. At the equivalence point, the conjugate base is the only remaining species from the weak acid. The hydroxide ion from the strong base continues to be added as the volume of the titrant increases. The weak acid has been completely consumed, and strong bases, remember, have negligible conjugate acids. Titrations involving a weak base and a strong acid. When a weak base is neutralized by a strong acid, the graph varies in a few ways. The equivalence point does not equal a pH of 7. It will be less than a pH of 7. So we can see here that the pH at the equivalence point is 5. A buffer region exists as the equivalence point is approached, and we can see that in the region of the graph right here. The half equivalence point is when half of the weak base is neutralized. So the equivalence point occurs at 25 milliliters, where the concentration of the weak base is completely neutralized. We can see that right here. Half equivalence occurs at 12.5 milliliters, where half of the weak base is neutralized, and we can see that right here. For weak bases, at the half equivalence point, the moles or molarity of the weak base is equal to the moles or molarity of the conjugate acid. Therefore, at half volume of the equivalence point, again, half equivalence, pH is going to equal 14 minus the pKb for the weak base. So if we look at this version of the Henderson-Hasselbalch equation, which has been rewritten to reflect pOH and not pH, we have pOH is equal to pKb plus the log of the concentration of the weak acid over the concentration of the weak base. when these two concentrations are equal to each other, this portion of the equation is removed and pOH is equal to pKb, as we can see right here. So because pOH is equal to pKb, to find pH, we can take 14 minus the pKb, which is the pOH, and find pH. The area around the half equivalence point is called the buffer region, since the weak base and the weak conjugate acid are both present in solution. At volumes less than the half equivalence point, the weak base is the primary species, and we can see that on the graph right here. At volumes greater than the half equivalence point, the conjugate acid, BH plus 1, is the dominant species, and we can see that on the graph right here. So the BH plus 1 is greater than the concentration of the weak base. At the equivalence point, the conjugate acid is the only remaining species from the weak base. The hydrogen ion from the strong acid continues to be added as the volume of the titrant increases. The weak base has been completely consumed, and strong acids have negligible conjugate bases. Sketch a titration curve for the titration of a weak base by a strong acid. The reaction for the titration is B plus H plus 1 yields BH plus 1. This is the titration curve that we're going to sketch to the best of your ability on the graph that's in your notes. Notice that there's 2 green points that you're going to want to add. So let's label the following points on the curve. The first point that we're going to label is the equivalence point. And the equivalence point is located at this green dot. So it is at a pH of 5. And this is the point where the hydrogen ion concentration equals the hydroxide ion concentration. This will be the steepest point in the graph. The next point to label is where pOH is equal to pKb. That is located at this green dot, half equivalence, where the concentration of the weak base is equal to the concentration of the weak acid. So pOH is equal to pKb. The next section is where pH is dependent on the concentration of the weak acid. That would be located right at the equivalence point, where all of the weak base has been neutralized. The next section is the region of maximum buffering. That is located in this section of the graph right here. The next point is where pH only depends on the concentration of the weak base. That's located at the very beginning of our titration curve. And then finally, pH depends only on the amount of excess strong acid. That's going to be located in this area down here. Next, let's sketch a titration curve for the titration of a weak acid by a strong base. The reaction for the titration is HA plus OH minus 1 yields H2O plus A minus 1. This is our curve for our weak acid. Draw it to the best of your ability. Then we're going to label the points on the curve. The first point that we're going to label is the equivalence point, and that point is located at this green dot right here, where the pH is equal to 9 and the concentration of the hydrogen ions equals the concentration of the hydroxide ions. The next point is pH is equal to pKa. We know that's going to be this green point right here, where we are at half equivalence and the concentration of the weak acid is equal to the concentration of the weak base. The next point is where pH is dependent on the concentration of the weak base. That's located right at this point of equivalence, where all of the weak acid has been consumed, and the only remaining part of the weak acid is the conjugate base. The region of maximum buffering is located in this region of the graph. pH only depends on the concentration of the weak acid. That's going to be at the very beginning of our titration curve. And pH depends only on the amount of excess strong base. That's going to be located in this area of the graph right here. Titration of a weak diprotic acid with a strong base. When a weak diprotic acid, example H2C2O4, where more than one labile proton exists, is titrated, there are two equivalence points. The curve is not as distinct because of the various proton donors and proton acceptors in solution. So this is the area where the first hydrogen of the acid is removed, and this is the area where the second hydrogen of the acid is removed. So what did you learn? We looked at titrations involving a weak acid and a strong base, titrations involving a weak base and a strong acid. We sketched a titration curve involving a weak base and a strong acid. We sketched a titration curve looking at a weak acid and a strong base. And then finally, we talked briefly about titrations of weak diprotic acids with a strong base. Need more help? Feel free to contact me. Have a great day.